Hi, my name is Natalie Graham, and I'm a graduate student working with Rosemary Gillespie. And my graduate work has taken me to the beautiful rainforests of Hawaii. Now, the reason why I work in Hawaii isn't just for sun and fun, it's so that we can study the biodiversity that's there, and particularly the biodiversity of arthropods, so insects, spiders, mites, all of those things. And we're interested in trying to understand how biodiversity changes over time. Hawaii is a great system for studying change over time because the islands are in a chronosequence. So each island is formed over a hotspot volcano. And because it's formed over a hotspot volcano and there's tectonic plate movement, the tectonic plates move and then each island is successively younger. So the island of Kauai is about 5 million years old. And then the big island, there's still lava forming. So we can look at communities of different ages by capturing all of the DNA from the arthropods on the island. So for my dissertation, I went and collected at 14 different sites on 4 islands, all of the arthropods, and I collected them from beating samples, so off of particular plants. So now I have an association of what arthropods are feeding on those plants, are doing predation on those plants, and I can build ecological networks and try to understand how these biotic interactions are also changing over time. So it's really interesting work. I'm using a new molecular method called metabarcoding, where the sample is collected in bulk, and then I can process the sample from DNA extraction all the way through to analysis as a complete sample all in one. And I can look at things like the genetic signature of how communities are changing over time. So for example, we would expect that on the very young sites, anything that is there as a part of that community has evolved somewhere else. So it evolved either some other parts of the older islands, or maybe it's a member of the community that has even come in from outside because of it's a non-native species. So we wouldn't expect them to be very genetically similar to one another. But as we go through time, we have some community properties that are changing and shifting. You have competition that becomes important, and competition leads to niche differentiation, where you have finer and finer scale of relationships between these species in the community. So eventually you end up with more of these tightly co-evolved relationships where organisms for millions of years have been feeding on each other, or using those plants as resources. And so we can actually see that signature of change in the genetic structure of the community. So this is really fun, interesting work just in and of itself, but it also can help us to extrapolate, to try to understand how communities are changing through time now, elsewhere, outside of Hawaii. So we can start to think, what properties of a community make it more stable, what make it more resilient to change. So for those introduced species, how are those introduced species affecting communities? And trying to understand just in general what the pressures of global biotic change are affecting communities overall. So that's why this work is important, and it's fun and interesting to do. And I've been so lucky to be able to be a part of ESPM and to have this broad knowledge that also is helping to preserve our environment. Thank you.